glial cells provide the structure for the cortex to form on. Neuroblasts, immature neurons, use the radial glial cells to guide them to their correct location. Once the neuroblast finishes migrating, the axons and dendrites, together called neurites, begin to extend. At the tip of the neurites is a growth cone. The growth cone has a flat undulating membrane called lamellipodia. Lamellipodia have spikes moving in and out to probe and move the axon called filopodia. The filopodia are drawn forward towards the midline by chemoattractants. Once the axon has reached the midline, the axon produces roboreceptors. The roboreceptors interact with the chemorepellent that pushes the axon away from the midline, continuing its growth. Once the axon is established, the neurons are ready to fire. There are two parts to a neuron firing, a chemical component and an electrical component. In the chemical process, neurotransmitter is released from vesicles in the tip of the axon. The vesicles merge with the neuron's membrane, freeing the neurotransmitter into the synaptic cleft. The neurotransmitter binds to a receptor on the dendrite of another neuron. The neurotransmitter binding triggers an electrical action potential in which voltage-gated sodium channels open on the axon and sodium flows in. The sodium ions diffuse down the axon, allowing more sodium channels to open. Positive influx causes voltage-gated potassium channels to open. Potassium ions rush out of the axon. This change in electric potential closes the sodium channels. The sodium-potassium pump uses ATP to restore the balance of ions. The action potential down the axon triggers the release of neurotransmitter into the next synaptic cleft. This triggers another action potential. These action potentials and neurotransmitter bindings move information between the body and the brain, allowing the body to move, think, breathe, and more.